Hello everyone, myself Dr. Ramachandra from IIIT Hyderabad. In this video, I'm going to share one of the tools related to hyperspectral. What we have developed as a part of our project on hyperspectral big data. We named the tool as a SEMUL, a semi-empirical model for upscaling mixed spectra. So this tool basically developed for the forestry studies as a part of forestry Canopy Upscale. And the team here are two students, Janice Sundar from myself from IIIT and Dr. Ramarao from IIT Prevent. Before I start discussing about this free Canopy upscaling model, I would like to give a brief introduction actually why this kind of Canopy upscaling model is required. Normally, we see geospatial technology, satellite remote sensing used in different fields, in forestry, geology, in urban studies. And if you see in reference to forestry studies, typically we have varied multispectral as well as hyperspectral data sets. Even there are some studies on using thermal remote sensing data sets. But if you see with reference to optical remote sensing, they have a good spatial resolution, but actually it is difficult to differentiate objects at a very final level. Suppose if you want to distinguish between different soil types or different rock types, or in case of forestry between different tree types, it is not possible using multispectral data sets as their spectral resolution is low. So when the data is collected at a very narrow spectral bands, what we are referring to as a hyperspectral remote sensing, it is possible for us to differentiate the objects based upon the subtle differences what actually we are going to see in these objects. So in such scenario, there is a need for these hyperspectral data sets to identify the features. And when you talk about hyperspectral things, we have varied spectral libraries. Hyperspectral data sets actually now currently we don't have any space-borne satellites, and getting data from airborne is very challenging and expensive. So now whatever the data sets that are created, they are stored in the form of spectral libraries, either for the soils or for the rocks or for the forest tree species or for any in fact any study of interest. So there are different spectral libraries are available to capture the scenario. So with reference to forestry things, where actually there are different studies on collection of spectra for different species. So all these data sets, what we are collecting, it is put into a vast spectral library. So people can refer to such spectral libraries, and then they can do the image processing or the analysis of satellite data. Since hyperspectral data is acquisition is challenging, as well as the analysis is also difficult, and especially data sets are collected from the ground using spectroradiometers. So in fact, everything appears to be challenging and expensive mode. So by the way of spectral library, we are trying to provide some kind of signatures or the information to the group of people where actually they can do their own analysis when the satellite data is available. So in this context, actually the project started for the development of the spectral database, and towards this actually in the project on hyperspectral big data sets, we started working on this canopy density, canopy upscaling model. So normally, if you refer to the forestry, uh, data can be collected from forestry to identify the density using multispectral data sets, mostly pan data sets, pan chromatic data sets are used for identifying the density types within the forest. However, that actually will not give us a clear picture about the health of the forest. So now, in order to see actually the biochemistry of the place, there is a need to have this kind of canopy upscaling models. So normally, in the process of obtaining the biochemical properties, earlier researchers they started using single leaf as a database collection. The spectra from the single leaf or spectra from the bunch of leaf, they are calling it this part as a canopy. So now, when this single leaf or the bunch of leaves they are treating as a canopy, and when the biochemical analysis is done for these leaves, they are actually calling this as a or correlating the bunch of leaf biochemical properties with that of the canopy. However, it is not actually a correct process because the type of environment what we see in the canopy, it is something different from the actually what we see in the lower parts of the tree. And in the ground, actually, it is very difficult to collect tree leaves because of the terrain of the forest, because of the height of the forest. So now people started upscaling these spectra that is derived from the bunch of leaves to the canopy level. And in this process, actually, varied canopy upscaling models are started coming up. So there are different canopy upscaling models. One is with the leaf reflectance models or one with the canopy reflectance models. Typically, we have something called different models like ProSail, Flight, Liberty, Sail, and currently there is a 3D canopy structure called GARD. So all these models are proposed globally to upscale the ground leaf spectra collected in the field to the top canopy level so that it can be matched with the spectra that is actually derived from satellite data. So the advantage of upscaling is that we are trying to upscale the biochemical properties observed from the ground to the top level. So in this process, these models are proposed. However, each model has its own drawback related to because there are most of these models are having many parameters are assumed. So based upon the assumption, actually these models may not be that much accurate. Then people started working with to get a more reliable data. So as a process of this, they try to retune the parameters. So all these actually models are rate of transfer models where actually we take some as a constant and some as a already fixed. And these canopy upscaling models are actually globally research from the global researchers. And the SEMUL model, what actually we created, is the first and unique for the within the country, as well as for the it is it's more superior compared with the other models because this model does not consider more or any much assumptions or the parameters. Whatever the parameters that are given for this upscaling model, all are derived from the field or from the lab analysis. So I'll just show you the process of creation of this canopy upscaling model. Basically, what are the requirements? It requires field survey data. So we need a tree species spectra and a tree biophysical parameters. Then whatever the leaves that are collected from the lab, they are subjected to lab analysis to get the biochemical parameters. And whatever the spectra that is actually upscaled using this model is actually compared with the satellite derived data to see how far the model is accurate, showing similar kind of spectral pattern like that of satellite data. So initially the process is, so we have chosen a study area as a Araku. The satellite data used here is CRISP proba data, CRISP data sets. It's a part of Araku, and the landscape has a different types, like with a good vegetation or something. There is a trees with the grass, and there are the different terrains with the different tall trees. So in such kind of scenario, it's very difficult to get the leaves of the top canopy. So what we'll try to do, we'll try to pick up from the lower canopy part of the tree, and these leaves actually we analyze, and finally the spectra from these leaves are actually upscaled to the top canopy level. So for this study, we have chosen 10 species based upon their economic and medicinal uses, as well as their dominance within the study area. So initially, a survey was carried out to see what is the dominance of these species in the study area, along with the suggestions from the forest department. So we have selected these 10 species. So each species has its own use, economic as well as the medicinal use. So now this spectra, what actually we have created, it is uploaded in a national portal called NISA portal, from where actually the one who are interested to study on any kind of these species, they can get actually metadata as well as the spectra for these species. So first thing is collection of the field data. So initially we collected the leaf spectra from ground for 3 different seasons, because the purpose is to see how a typical single tree behaves in different seasons. So we have chosen wet season, the dry season, as well as a regular normal season, where we try to see how these leaves vary, their biochemical constituents vary, and based upon their biochemical properties, how the leaf reflection pattern is changing. So whatever the data we are collecting, so data was collected using ASD spectroradiometer with ranging from spectra from 400 to 2500 nanometers. So we collected a bunch of leaves where actually we averaged all the, we took 10 readings and we averaged it, making as a single spectrum. However, whatever the method we are choosing for the collection, there is always possibility of having some kind of noise or the atmospheric interference. So whatever the spectra that actually we have collected from the field, it shows actually so much of noise within the spectrum. So first part is to process the spectrum. In processing this, most of the studies of researchers, they are actually using either a Fourier transform method or the spline method to make the curve as a smooth. However, when you do such kind of spline or the Fourier transform method, we are losing actually all the small dips and the peaks, which are typically essential for the identification of different species. So we are able to identify one species different from the other based upon these peaks and the troughs. So we did not choose the kind of FTR any spline method. Here actually we created our own method. We call it as a signal noise ratio method, where we try to remove such noise. So we have, this is the collected spectrum that is derived from our own method. So this is where, in this way, we have created spectra for all the 10 species in different seasons of winter, monsoon and summer. So this part actually is the processing of the spectral data sets. Next part is the biophysical properties. So when we are collecting the leaf spectrum from the leaf, at the same time we also calculated a leaf area index using plant canopy analyzer. This is one of the biophysical parameters that actually we are going to consider in our model. Then we have third is the biochemical properties. So for all the species what we have collected, actually they are dried. So we took the weight of the leaves before and after the drying. Then again they are weighed and they are subjected to extract different biochemical properties like cellulose, hemicellulose and lignin. Chlorophyll measurement, estimation of the protein, what is the protein content within the leaf, as well as the nitrogen estimation using Dildal method. So now once these 3 parameters are obtained, that is the spectra, biophysical properties and biochemical properties. We created a flow where actually we integrated all these components to upscale the model. So here you can see leaf biochemical parameters. Basically, these are the four parameters we are considering: chlorophyll, nitrogen, lignin and cellulose, and the water content. Then the canopy properties, basically we make use of leaf area index, and the spectrum, the processor spectrum after removal of dropouts and noise from the data from the spectrum, whatever the spectrum that is there. We also try to differentiate different kinds of species spectra using different similarity and dissimilarity tests. So fine tuning what is the spectral property. So that is actually forming the one of the spectral library of the leaf. However, this leaf spectral library we have to upscale to the canopy level. So then these biochemical properties, biophysical properties, and the leaf spectrum, they are finally integrated in the model and we process it. So whatever the canopy level reflectance, in between actually we use different mathematical models, formulas to upscale the ground level parameters to the canopy level. Ground level chlorophyll to the canopy level chlorophyll. So different formulas are available to calculate chlorophyll, nitrogen, protein content, and lignin from the literature. So all these are integrated, these parameters, and finally we created a canopy level reflection spectrum. So this spectrum actually finally going to the canopy level spectral library. So before going to this canopy level spectral library, there is actually need to validate actually what is the spectrum that is created by us. So this is actually the output that it is created in Python. So there is an option to give a single input. You can choose the leaf spectrum file, or you can also have the batch input. At a time you can give all the spectra for the modeling. So column number where actually the reflectance values are there, that value actually we have to indicate here. Then we take different parameters like what is the fresh chlorophyll content. So this chlorophyll content, which is in the percentage, it is calculated in the field for the chlorophyll estimation instrument. Then nitrogen, lignin, and they are coming from the biochemical analysis along with what is the fresh leaf weight. Then we also have dry chlorophyll content, that is post drying of the leaf materials, what is the protein content and what is the cellulose along with the dry leaf. This para basically consists about the biochemical properties. So here the canopy properties, the biophysical properties, what you are giving is a leaf area index of that particular tree species. So now once all these parameters are given, we can choose how we want to see the spectrum. So whether you want to see only the upscale spectrum or the one method spline method, because people also argue that the spline method and the FT, Fourier transform method also are useful. So to show that we also created a spline model, and normally without water spectrum. So normally if you see spectra, actually it is discontinuous. When you add water thing, actually we can see the continuous spectra. Whenever there is no water, we can see it as a discontinuous. Or you can want to see all, you can choose. So any of these option you can choose, and finally you can store it. But actually you can have the show and create option, or just to show or create. So this model, for example, so we have given one file. So one this is the local name of the species. Then actually what is the column name and what are the biochemical properties that are actually derived from the lab, as well as what is the biophysical property, that is the leaf area index. And finally, you can see the spectrum. We have the blue one is the leaf spectrum, and the green one what we are seeing is the canopy reflectance. This is a leaf reflectance and upscale reflectance without water region. And this actually you can see all the four leaf reflectance, canopy reflectance, spline, as well as the reflectance without water. So obviously what we can see here is the green one is the normal upscaled one, The leaf reflectance is more or less is high because the conditions what we are seeing at the ground is something different from that of the canopy, where actually there is a different illumination effects as well as a crown effect. So then we compared actually our leaf spectrum with that of the upscaled canopy spectrum, where actually we try to see whether what kind of pattern we see. So we see that leaf spectra and the canopy spectra differences is evident in the form of different in their magnitude of the spectral reflectance in specific regions of the spectral region and the direction of the change of the reflectance across the spectral region. So for some species actually there is a difference is identified only in the magnitude of the upscaled reflectance. If you see some species like Polyalthea, here actually there is something anomaly. This is mainly because of the leaf structure. So the leaf what actually we collected during the winter season, it is actually the cell membrane is damaged. There is a more actually porous. So we find that kind of different abnormality within the spectrum. Obviously, this is the thing we want to capture because the study want to see how the spectrum behaves with the under different stress conditions. We also compared our upscale reflectance with that of the spectra that is derived from the satellite data, CRISP proba. So we have ground locations. We have picked up 5 locations from the ground where actually we know the presence of the tree, and we try to see what is the difference between the upscale reflectance with that of the pixels spectra that is obtained from the satellite data. So we can see that actually the upscale canopy and spectral spectra match with the most of the spectral angular distance being less than typical less than threshold of 5. So we managed with found that typically the threshold of 5 at this time we can see a good matching between the spectra that is coming from the satellite data and that of the upscaled thing. So further details of this actually we can see in this paper, semi-empirical model for upscaling leaf spectra. This is available online, and if at all anybody needs, you are not able to find this paper, please write to me. Thank you. So please write to me at this mail ID.